Hello, everyone. My name is Luisa Schulte, and I work for Maanaki Whenua Landcare Research. And since July the 1st, we're actually part of the Bioeconomy Science Institute. You might have heard we merged with Plant and Food Ag Research and Science. But anyway, I just want to give you an update on the biocontrol side of things, mostly about Chilean Needle Grass, and I will touch a little bit on the Nasella project as well, but I haven't worked on it myself, so it will be quite short. So I want to give you an overview of the two different methods we have in biocontrol. One is classical biocontrol and the other augmentative. So I shortly want to explain this and then tell you about the biology of the Chilean needle grass rust, how it actually damages the grass, give you a short overview about the history of the Biocontrol project because it has been going on for quite a while, and then tell you about the pieces that we made this year, last year actually. And the last thing is about the Nasella project. But I thought I maybe just shortly introduce myself. I'm from Germany, but since September 2023, I work with Maanaki Whenua in Auckland. So yeah, I've been only around, not even 2 years. So if I don't know all the ins and outs and cannot answer all your questions, please be kind to me, but I will try very hard. Yeah, and before that, I just did lots of biology stuff, genetics, and a bit of mixture of genetics and ecology. So I haven't worked in biocontrol before coming here, but yeah, I plunged myself into it, and but I'm still in a learning curve, so to say. But yeah, classical biocontrol. The method is based on the so-called enemy release hypothesis. So the idea is when a new plant gets when a plant gets transported into a new area where it hasn't been before, it often comes, so to say, alone, naked, maybe just as a seed. And it doesn't bring with it all the natural enemies, all the insects and diseases that keep it under control in its home range. And in biocontrol, we want to use those natural enemies, which can be insects, mites, or pathogens like fungi or bacteria, and take them from the home range of the weed and bring them into the range where the weed has been introduced. And the idea is that once we bring them over, they can establish, they can disperse, they're living organisms, they will replicate, and they will help to control the weed in the introduced range. But because we want to bring a living organism into a new country, there's a lot of risk assessment needed. So we have to do a lot of testing, so to make sure that no native plant or valuable crop will be affected by the new organism that we want to bring in. And biocontrol is quite slow. Normally, even when we do the release, it's not a quick fix. These are living organisms. They need to build up populations, and it takes a while. Sometimes some biocontrol agents are quick, but it's not like the usual case. But yeah, once it is actually established, it will stay forever, basically. A new organism that is brought into the country and hopefully helps to control the weed forever. High initial investment. It takes a couple of years to do those research projects, but cost effective over time if successful. Classical or importation biocontrol involves surveys in the home range of the weed, and this is a bit. We can get rid of this. Host specificity testing. So we test the agents and see that they don't touch valuable plant species. And lots of approval and bureaucratic work and to get permission to get it into the country and release it. Just some expectation management. So biocontrol cannot eradicate a weed. It's not really the goal of biocontrol, because these are living organisms. When a weed gets really rare, or these organisms will not find the weed anymore, so the population of the biocontrol agent will go down, and if the weed comes up again, then in theory the biocontrol agent population comes up again, and it's more of an organic thing. And the goal is more to get the population of the weed to a level where it's not longer as damaging as before. And yeah, as I mentioned, it's not a quick fix. Unfortunately, it takes a while for these organisms to build up levels where they can effectively provide control. And so, yeah, the most important thing is when we release a biocontrol agent on a property, it should not be sprayed, because you kill the plant, you kill the biocontrol agent, and it cannot establish. And yeah, an example of classical biocontrol is, of course, our Chilean Needle Grass rust. So the plant comes from South America, is introduced to New Zealand, where this plant is, grows better and more prolific and makes more problems because it has no natural enemies. And so, surveys are done in the home range of the weed, in this case in Argentina, and they found a rust that is damaging to the grass, and did a lot of testing in Argentina. Testing can also be done in New Zealand, but back then we didn't have a facility that was able to contain rusts, because they form spores and you have to be very careful. So now we have a containment facility in Auckland that can handle rust. So we have filters and negative pressure, and lots of security, so the rust doesn't go out before we have actually approval to release it. But most of the tests were done in Argentina in this case. Then the idea is, you bring it over into the new range and release it. But I will talk more about this in a minute. First, I want to tell you a little bit about augmentative or inundative biocontrol. That's a different method. And in this method, we use native or already present agents, so present in the range where the weed is present, so where this introduced plant is present. It can also be insects, mites, or pathogens, so fungi or bacteria. They are present, but they're obviously not damaging enough. They don't control the weed, so the idea is we could mass produce them and then release them, and by these means control the weed, because maybe the climate is not quite right, or the conditions are not quite right, but if we help this organism a little bit, mass produce it, release it all at once, it can effectively control the weed. So it's more of a short-term, and seasonal control needs to be repeated, because the agents are not expected to persist long-term or build up big populations over a long time, but they have to be reapplied. But because the agents that we use for this are already present, the host specificity requirements are less stringent. So it doesn't need to only affect one single weed, but can also affect maybe some native plants, but that are not in the direct surroundings where it is applied. And when we use microbial agents, so fungi or bacteria, but mostly it's fungi, we call those augmentative biological control agents. We call them bioherbicides. And so bioherbicides, they work just as a herbicide, but they contain a living organism. Can also contain only compounds derived from them, but mostly they contain actual living organism. So the development of a bioherbicide also takes pathogenicity testing. So you look for strains that are very, very damaging, but still specific. Then you want to design and optimize the formulation so it's effective, and then it's applied basically like a herbicide. So you might think, okay, if it's like a herbicide, why not use a herbicide in the first place, since it can actually kill a weed effectively, but a bioherbicide has some very good advantages. And the biggest is that it's specific to the weed. So you could spray it over the whole paddock, and only your weed would be affected, and all the other plants grow happily beside it. And yeah, it's also safe for the environment. It's not toxic, so it doesn't pollute waterways, and yeah, doesn't harm humans or animals. And with herbicides, you also get the problems of building up resistance in plants. So with bioherbicides, that's much, much less likely, because they have a really complex mode of action, because they're living organisms. So yeah, this is called augmentative or integrative biocontrol. And an example for this is the work that has been done on Nasella tussock. So once again, the weed is native to South America, but the agents, they were actually looked for in New Zealand. So surveys were undertaken in New Zealand, and also the testing and the formulation work was done in New Zealand. So it's a little bit, two different methods. Okay, any questions to that? I can also take questions at the end. Okay. So now to the Chilean needle grass rust. So just want to talk a little bit to the biology of the rust. So these are the spores. That's the way it disperses. Wind disperses these spores, and if one spore lands on a leaf of Chilean needle grass, and we have some moisture around, that's very critical. The fungus always needs some moisture to germinate, 6 or 8 hours. If that's the case, then it can germinate, and it penetrates the leaf through the little leaf openings on the surface of the leaf, and then it grows inside the leaf and lives there and takes up energy from the plant. And then after a while, if it gets enough energy from the plant, it forms these pustules, and they break through the surface of the leaf and form lots of new spores. And these spores are then again dispersed by the wind, and the cycle can start again. And so this is how the rust damages Chilean needle grass. With the right conditions, it can go through the cycle many times in one season. So if it's a nice and wet season, that's ideal. So it would be like in spring or autumn here, and not so much in summer. Yeah, if it infects Chilean needle grass, the leaves, they often die prematurely. And what was observed in Argentina, where this rust comes from, is that infected plants produce much, much less seeds than healthy plants. So that's what we hope to see here as well, hopefully. A little bit about the history of the project. I've been here only the last 2 years, but you guys have been here maybe for the whole story. So this project goes way, way back. It started in 1999 with Australia launching the project. But I really just want to acknowledge the people that have worked mostly on this. And first and foremost, this is Freda Andersen, our collaborator from Argentina. She spent like half of her work life on Chilean needle grass, and also she worked also on Nasella tussock, trying to find damaging fungi and overcoming lots and lots of obstacles. And the biggest one was get permission to export the rust. It took 10 years because of some law changes. I don't want to go into details, but she persisted, and she managed to get the rust out of Argentina. And so we got it here, and we finally could do our first releases in 2024. I just also want to acknowledge Jane Barton. She was a plant pathologist in Auckland before I came along, and Alana Dembrain, you might have met her. She gave a talk at the last of your meetings. She left us last year. She went back to South Africa. Yeah, so as I mentioned, in October 24, we finally, after more than 20 years, did the first release. And we did the release at 3 sites here in Blind River. And we selected 10 plants per site and 10 leaves per plant. And yeah, this is how it works. So basically, I harvested the rust spores from our facility in Auckland, mixed them with white talcum powder, and then just brush it on the leaves. And then we spray it with water and packed it into a plastic bag so it retains the moisture for a while. And yeah, I did this together with Brent Hans from Marlborough Council, and he went back to the sites then in spring, and at one site, actually the site from Tim Strathers, I could validate that the rust had established, and I found the spores on the leaf samples. But yeah, we still have to see how it develops and if it will spread from the site, and yeah, if it can establish over time. Yeah, I also did one release in Canterbury in Spotswood, and also that also did establish, or I could recover the spores in spring. But yeah, I would love to see some more samples next spring and see whether or not they're still there. Yeah, the next steps for this project, more releases by Marlborough District Council. And yeah, I'm also involved in a project with Australia. So obviously they also got Chilean Needle Grass. The problem is that they have 2 native grasses that are susceptible to the rust, or that's what was found by the tests done in Argentina. And yeah, and so what I am currently doing is I have the Australian grasses in our containment in Auckland, and I'm infecting them and see whether they also develop pustules when we have spore concentrations that are more similar to the natural concentrations. Because when we do the tests, we basically we overload them with spores, and so it's not really a natural condition. Yeah, but I'm still testing. Yeah, and if we see that this rust is successful in controlling the grass here, then we might look into new strains from Argentina to also control the Hawke's Bay population, because unfortunately this rust is so specific that it doesn't even go in the Hawke's Bay population, which is very unfortunate. Yeah, that's what I've got to Chilean Needle Grass. Oh yeah, just wanted to reiterate, if you have a release on your property, please don't spray, because yeah, you kill the grass, you kill the rust. Yeah, the Nasella Tussock project, unfortunately not so much or nothing has been done since your last meeting. But I just really capture what has been done. So Freda had also looked for a classical biocontrol agent in Argentina, but nothing suitable was found. Some fungi for bioherbicide development were found in New Zealand and Australia, and the most promising agent is called Tiarosporella graminis, what Seona Casonato called Black Death. So Seona Casonato was the person that worked on it for the last project, the last couple of years. Yeah, this agent causes tiller death and reduced flowering and viability of seeds. Unfortunately, yeah, Seona went back. She worked at Lincoln University and then went back to Australia. And people from Ag Research wanted to pick up the work and put together a project application, also together with Alana and Brian, who worked with Maanaki Whenua, but it didn't get funding. But it is, MPI would have funded the project, but they wanted some co-funding, and I couldn't find the co-funding. And this is very unfortunately what happens very often to a science project. You put a lot of work into it, you do research for a couple of years, and then money stops and people go away. And yeah, so unfortunate. I mean, a lot of has been done. There are still some things that need to be done in order to get it working. So this is develop a stable and efficacious formulation, safety testing, so to make sure the pasture plants wouldn't be affected, field trials and refinement of the formulation, then develop techniques for mass production application. And this is optional, but theoretically for a big market like Australia, you could also look at commercialization. Yeah. Yeah, I don't know. This is something maybe for discussion. I mean, I would be interested in picking up the work, but I also would need to get funding. I talked to the people from Ag Research. They also said, yeah, we would be interested if we can find some money. Yeah, but funding for science at the moment is not great. So yeah, money is tight everywhere. But yeah, that's the current status. And that's all from my side. Happy to take any questions that are some.
Can I ask you to go back to the Chilean Needle Grass? And you talked about the spread from the rust spread here. Is there any potential with the seed to transfer or take rust with it as it's moved, or not?
Not really. It stays actually in the leaves. I have never seen any pustules on the seeds. And,
How robust are those spores? Will they handle the rainfall event and get washed down the creeks and reinfest needlegrass down the creek? I'm just thinking of the speed to spread it quickly.
Well, then it would somehow need to get from the creek on the leaf.
Yeah,
so it's a bit. I'm not sure if that would work out because.
In a flood event, it's washing across needlegrass.
Yeah, maybe. But basically, the spore needs to land on the leaf surface, and then it has like 8 hours to grow a little germ tube and find a little leaf opening, and that's it. I mean, the way it works is that it just produces heaps of spores, and one of them will somehow, maybe, hopefully, land on a leaf and can do an infection. But most of the spores just don't do anything. But yeah, a flooding event might. I'm not sure, but it would be interesting to find out.
Nature finds a way, normally.
So are you looking for properties to put their hands up for releases? Or have you got enough already?
So yeah, maybe Brent can.
We do seek and release later in the year.
Yeah, so ideal are properties that have an area with Chilean needle grass that is a bit shaded, moist if possible. So not a north-facing slope, but rather something with a bit of shade, and most importantly, that it's not sprayed or braced, because yeah, we need nice and healthy plants. I know you don't want nice and healthy plants, but it's ideal for the rust.
And obviously with Blind River and the train track running through the middle, there's the fire risk as well. So we want to try and get them spread out a little bit just before I kick off. We know how dry it gets in that area with the train going through. It's a bit of a risk that we invest all in the same little spots.
You mass produce the spores. So needlegrass doesn't produce spores, so that we can do some extensive releases rather than mucking around a little bit.
Yeah, I mean, that's what I basically do in our greenhouses up in Auckland, but
small scale.
Yeah, very small scale. It's a lot of work because yeah, unfortunately, these rusts, you cannot grow them in media or like liquids or stuff. It has to be in the plant. It only can live in the plant. So what I do is take a little brush the spores on these, keep them moist, and then come back with a little kind of I have like a vacuum, and I vacuum them off. Yeah, it's a lot of work, but the idea is that it then can self-replicate. So you do a couple of releases, but then it will take off if the season.
All right, any more questions? Well, I'm sure people will chat to you, especially all those people with lots of change in their pocket. Awesome.